Hello once again. Now we are dealing with partial linkage, which is the most probable case that will be in your tests. The case of dihybridism partial linkage test cross in cis position will give such this example. If we cross a hybrid Drosophila having dominant phenotype of brown eyes and brown body with a birecessive one having red eyes with orange body will give, in this example, we have 30 Drosophila having four unequal proportions in the offsprings. Four unequal proportions, too high of them and the other is too low. The characteristics of the hybrid parent will be found in high proportion if it is the case of cis. So the Drosophila and the offsprings which are similar to the parent, this parent, the male hybrid one, are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. So we have 12 Drosophila having the dominant phenotypes. Dominant phenotypes having brown eyes and brown body. And the other 12 Drosophila will have the genotype of the birecessive parent, which is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. While the two low, which are the results of recombination, which is an intrachromosomic recombination, are three Drosophila having the dominant trait or the dominant trait of the eyes and the recessive one for the body, which is a 1, uh, what else? Where's the others? Where are 2 and 3. On the other hand, the other three ones which constitute the low proportion having the dominant phenotype for the body and the recessive phenotype for the eyes can be this red eyes with brown body, I 1, and there would be existing two other Drosophila having the same phenotype. This is 1. What else? Red Drosophila with 2 and the third one here. Okay, so the proportion will be 12, 12, 3, 3. These are the proportions, and if we sum up the the hybrid ones, or sorry, the recombinant ones, the recombinant Drosophila offsprings will be the Drosophila proportion, which have the least number of the offsprings, will be 3 plus 3 divided by the total number, which is 30, times 100. Then the recombination percentage in this cross will be 20%. So this test cross in the partial linkage cis position case will have 20% of recombination. And consequently, this 20% recombination tells us that these genes are located on the same chromosome with a distance of 20 centimorgan between them on the same chromosome. Okay, so this percentage of recombination indicates the distance between the genes on the same chromosome. Also, this number will indicate the percentage of uh, gametes produced by the hybrid parent. So the hybrid parent will have 20% of recombinant gametes, then uh, his or its gametes will be 40% to other 40% and 10% for each of the recombinant chromosomes carried by the gametes. Later on, we will see an example and we will have more idea about how to calculate the percentages of gametes. Now, concerning the case of test cross-partial linkage in transposition, in this case, uh, a hybrid Drosophila is being crossed with a birecessive one, but the four unequal proportions in the Drosophila will be uh, the highest percentages will have one dominant and another recessive alleles are linked in the same proportion of the offsprings of the high frequency. So the highest frequency will be these no, these types of Drosophila, which join a recessive trait for the eyes and the dominant trait for the body. They constitute 12 of the offsprings. However, the dominant the dominant characters joined in the same individual will be the least number or the lowest frequency, which is completely opposite to that of the cis case. Okay, so this is the highest one. We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. Once again, this is the other type of phenotype with the highest frequency. The dominant characteristic for the eyes, which is brown, and the recessive characteristic or trait for the body, which is orange, having 1, 2, 3, 4, and till 12. Okay, so the case of transposition. In the case of transposition, we will see the highest frequency, and the offsprings will have one dominant allele expressed along with the recessive allele for the other gene, okay, which is opposite to that of the cis position. Now, as a summary, this summary will be must be memorized because it collects all the uh, expected results or percentages which will be produced by a pure cross, self cross, or test cross. You can take a screenshot for this one and memorize them. So usually the dihybridism case comes in the official exams. If the genes are independent, then the self cross will give 9331 and the test cross will give four equal results. However, in the absolute linkage case, the test cross will give in this position 50% to 50%, two halves of the offsprings. Each of them will carry the dominant phenotypes linked together in one individual, one proportion of the individuals, and the other 50% of the individuals and the offsprings will have both recessive alleles, which are linked together. However, in the transposition in absolute linkage, this results will be one dominant allele is linked to a recessive one, and the other 50% will show a recessive phenotype is linked to a dominant one. However, in the case of partial linkage, the test crosses will give too high and too low percentages in this manner. So if the high proportion of the offsprings carries dominant alleles together in the same offspring and the recessive alleles in the same high offspring, the other same high offspring, then it is the case of cis position in partial linkage. While in partial linkage transposition, the highly percentages in the offsprings will have a recessive allele for one of the traits and a dominant one in the same individual for the other trait. Okay, and finally, some notes should be memorized because uh, the applications on this chapter are somehow tricky. So, first of all, if F1 gives gametes in four equal proportions, then the genes are independent. Second one, if the F1 gives gametes, F1 means a hybrid individual or a heterozygous one, gives gametes of four unequal proportions, then the genes are partially linked. Third, if the F1 gives gametes of two equal proportions, then the genes are absolutely linked. Also, the other rules are the fourth, if the cross and its inverse gives different results for a given gene, then this gene is located on X chromosome. Fifth one, if the cross of its inverse gives if the cross and its inverse gives the same results for a given gene, then this gene is located on an autosome. The sixth one, in polytry, males have XX and females have XY gonosomes. The seventh one, lethal alleles kill their homozygous carriers. So lethal alleles are the alleles that kills only their homozygous carriers. The eighth note, when the variation involves only males offsprings, then the crossing over rate is calculated according to the males only. The crossing over rate is denoted by the percentage of recombination, which is the number of recombinant offsprings over the total. And in this case, as note 8, uh, the total is calculated from the males only. The ninth one, XY chromosomes are not homologous, so they can't undergo crossing over. So these chromosomes are the only ones that will not undergo crossing over, and intrachromosomic recombination will not take place in this case. The 10th note, when the gene is carried on X chromosome, then males' phenotypes reflects their genotypes. The 11th one, the parental offsprings indicates the pattern by which alleles are arranged in F1. Okay,